What is microalbuminuria? Which are the hematopoietic vitamin? What is the composition of ORS? Well, these were some brief answer questions which were asked in MUHS Supplementary Examination 2023. So let's see the solution to these BAQ along with other questions. So let's start. So let's start. Solution to MUHS Summer 2023 Supplementary Examination, Paper 2 Brief Answer Question. These were the all BAQs which were asked. We will see in detail the solutions to each of these. So first question is, that is question number 2A. What is Wobble hypothesis in genetic code? Mention its significance. So this question has two sub questions. First, you have to write what is Wobble hypothesis, and second, you have to write its significance. So 1 mark for each sub question. First, we will see the answer to first sub question, that is what is Wobble hypothesis. So Wobble hypothesis is a mechanism by which a transfer RNA can recognize more than one codon for a specific amino acid. Okay, so transfer RNA can recognize more than one codon for a specific amino acid. This is Wobble hypothesis. So according to it, the codon-anticodon pairing follows the traditional Watson-Crick base pair rules for first two bases of the codon, but it is less rigid or less stringent for the third base. Okay, so traditional Watson-Crick base pair rule is A with U and G with C. So this is followed by only first two bases of the codon, and it is less rigid or less stringent for the third base. Now, movement of first base of anticodon, which is 5' end of the anticodon, allows non-traditional base pairing with the last base of codon, that is 3' end of the codon. Okay, and this movement is called as Wobble. Okay, movement of first base of anticodon, which allows non-traditional base pairing with the last base of codon. This movement is called as Wobble. Okay, so for 1 mark, that is enough. That what is Wobble hypothesis. Now, what is its significance? It has two significance. First, it allows a single tRNA to recognize more than one codon, so that only 32 tRNAs are required to translate all 61 codons. So we do not require 61 tRNA. We do not require 61 tRNA for all 61 codon. Body needs only 32 tRNA, that is 31 for amino acid and 1 for initiation. Okay, so 32 tRNA, only 32 tRNAs are required for translation of all 61 codon. Okay, second significance is very important due to this loose base pairing between third base of codon and first base of anticodon. Transfer RNA can rapidly dissociate from its codon during translation. So rapid dissociation of tRNA during translation. So it will accelerate the translation process. Okay, so due to this, due to less rigidity, according to Wobble hypothesis, so tRNA can get rapidly dissociated during translation. So these are the two significance of Wobble hypothesis. Okay, so that is enough. You will get 2 out of 2 mark if you write this. What is Wobble hypothesis, and you have to mention its significance. Let's see the succeeding question. Question number 2B. Enumerate various active forms of vitamin A. Which form of vitamin A acts at gene level? Again, two sub questions are there. You have to enumerate the active forms of vitamin A, and you have to write which form of vitamin A acts at gene level. So what are the different forms of vitamin A? Aldehyde form, that is retinal, alcoholic form retinol, and acidic form, that is retinoic acid. Okay, and which form of vitamin A acts at gene level? Answer is retinoic acid. Very specific, that is retinoic acid. So there are 4 words you have to write and half mark for each word. So you will get 2 out of 2 if you write like this. There are 3 active forms of vitamin A: retinal, retinol, retinoic acid, and which form acts at a gene level, that is retinoic acid. So 4 component, half mark for each, 2 out of 2 mark. Let's move to the subsequent question. So question number 2C, write metabolic fate of essential sulfur containing amino acid. So this question is a twisted one because they have not asked directly the name of essential sulfur containing amino acid. So you should know what is the name of essential sulfur containing amino acid. Which are the sulfur containing amino acid? Methionine and cysteine. Out of these two, only methionine is an essential sulfur containing amino acid. So we have to write the metabolic fate of methionine, which is essential sulfur containing amino acid. So to answer this question, I have drawn a small pathway. So it will be better to write the answer of this question. So methionine is converted to S-adenosyl methionine, which is the active form of methionine, and this is the only methyl donor in the body. Enzyme required is methionine adenosyl transferase. Okay. Now what is the fate of SAM, that is S-adenosyl methionine is converted to S-adenosyl homocysteine with the help of enzyme methyl transferase, and the methyl group is donated. Okay, so any compound which require methyl group will accept that methyl group and get converted to methylated product. This reaction is called as transmethylation reaction. So this SAM is the methyl donor in the various transmethylation reactions which are occurring in our body. Now what happens to this S-adenosyl homocysteine? It gets converted to homocysteine. It will get lyse with the help of adenosyl homocysteinase. It gets lyse to homocysteine and adenosine. Now there are different fate of this homocysteine. It can get converted to succinyl CoA. Okay, it can get converted to succinyl CoA. There are different intermediate which are produced during this conversion of homocysteine to succinyl CoA, like alpha ketobutyrate, then propionyl CoA, then succinyl CoA. But as it is of 2 mark, question is of 2 mark, so you can skip this, but draw this arrow which represent that there are some intermediates. Now succinyl CoA can get channeled for production of glucose with the formation of oxaloacetate. Okay, again I have missed this. Okay, because it is only of 2 marks question. So succinyl CoA to glucose. So what it represent, that methionine is a glucogenic amino acid. Okay, methionine is a glucogenic amino acid. Other fate of homocysteine is that it gets converted to cysteine. Get converted to cysteine. Okay, now if you want to write the metabolic fate of methionine, the important metabolic fate of methionine, the first will be S-adenosyl methionine. Okay, because it is the active form of methionine and it is involved in various transmethylation reaction. It donates its methyl group to various compounds to form methylated products. So first metabolic compound which is important as a metabolic fate of methionine, first will be S-adenosyl methionine, that is SAM. Second will be homocysteine. Second will be homocysteine because again it has a different fate. So second important metabolic compound produced from methionine will be homocysteine. Third will be glucose, which represent that methionine is a glucogenic amino acid, and fourth will be conversion to cysteine. So fourth will be cysteine. So these are the important metabolic fates of methionine. Okay, so if the question is like this, that enumerate the metabolic fates of methionine. So you can write this 1 2 3 4, that is first is SAM, that is S-adenosyl methionine. Second will be homocysteine. Third is important, that is glucose, and fourth is cysteine. So these are the various metabolic fate of essential sulfur containing amino acid, which is methionine. Okay, let's see the succeeding question. Question number 2D. Define microalbuminuria. States its clinical significance. So 1 mark for each sub question again. Definition of microalbuminuria and clinical significance of it. So first, what is microalbuminuria? Excretion of albumin in urine in the range of 30 to 300 milligram per 24 hours. That is called as microalbuminuria. This is very important. This range is very important. 30 to 300 milligram per 24 hours. Okay, 30 to 300 milligram per 24 hours is called as microalbuminuria. What is its clinical significance? It is a screening test for early detection of microvascular renal disease. For example, in diabetic nephropathy, it is very important. Diabetic patients are screened through this microalbuminuria. So what is the clinical significance of microalbuminuria? That it is a screening test for early detection of microvascular renal disease, for example in diabetic nephropathy. Okay, so that is enough for writing 2 marks. You will get 2 out of 2 if you write like this. Okay, next question is 2E. Explain the role of atrial natriuretic peptides in water and electrolyte balance. Explain the role of atrial natriuretic peptide in water and electrolyte balance. You must have studied this ANP, role of ANP. So in the name it says that it is natriuretic. Natriuretic means promote the excretion of sodium. Promotes the excretion of sodium. So we will see the role of ANPs, atrial natriuretic peptides. So peptide acts on the kidney and increases the sodium excretion, as the name suggests. Okay, increase the excretion of sodium, and it also increases glomerular filtration rate, that is GFR. So ANP increases sodium excretion and GFR, that is first. Second role, how it is happening, how the sodium excretion is increasing? By inhibition of RAS, RAAS, that is renin angiotensin aldosterone system is inhibited by ANP. Okay, this system causes vasoconstriction. Okay, so this is inhibited by ANP. So this natriuretic action is due to inhibition of renin angiotensin aldosterone system. So obviously it will antagonize renal vasoconstriction. Next important part played by ANP is it increases the capillary permeability. Increases the capillary permeability. Due to this, it shifts fluid from intravascular to interstitial compartment. Okay, ANP shifts fluid from intravascular to interstitial compartment. How it is doing that? By increasing the permeability of the capillary. Okay, next point is it causes functional antagonism to all the modalities of humoral or autonomic induced vasoconstriction. So all modalities of humoral or autonomic induced vasoconstriction is inhibited. Okay, vasoconstriction, it can be by humoral or by autonomic mode. So that is inhibited by ANP. Okay, so that is enough for writing the answer for 2 marks. Okay, so this is the way by which ANP maintains water and electrolyte balance. So as the name suggests, natriuretics promote a sodium excretion. How? By inhibiting RAS, it antagonizes renal vasoconstriction. Second action, by increasing capillary permeability, it shifts fluid from intravascular to interstitial compartment. Okay, so let's see the subsequent question, that is question number 2F. Enumerate hematopoietic vitamins with one function of each. Okay, now you must have studied the vitamin chapter, but there may not be a separate section for hematopoietic vitamins 1, 2, 3, 4. Okay, you have to recollect what you have read. Okay, so I have provided the solution for this question, that is which are the hematopoietic vitamin. Now hematopoietic vitamins acts by increasing the DNA synthesis or by increasing the heme synthesis. So this is only of 2 mark. So let's see, first hematopoietic vitamin, very important, is folic acid. Active form of folic acid is tetrahydrofolic acid, which is required for conversion of deoxyuridine monophosphate to deoxythymidine monophosphate. Okay, so for conversion of DUMP to TMP, okay, DUMP to DTMP, there is requirement of tetrahydrofolic acid, which is the active form of folic acid. So if this step is inhibited, this step, that is conversion of DUMP to DTMP, that is with the help of thymidine synthase. If this step is inhibited, there will be no formation of deoxythymidine triphosphate, that is deoxy TTP will not be produced. So there will be the no DNA synthesis, so no RBC synthesis. Okay, so that is why it is hematopoietic vitamin. That is first. Now for action of folic acid, you require vitamin B12 because it releases free tetrahydrofolic acid from methyl tetrahydrofolic acid. Okay, so it is required for generation of free tetrahydrofolic acid from methyl tetrahydrofolic acid. If vitamin B12 is deficient, obviously a folic acid function will be hampered. Okay, and this is called as folate trap. Folate get trapped in the form of methyl tetrahydrofolate. So in B12 deficiency, you will observe folate trap. Okay, this is second hematopoietic vitamin. Third hematopoietic vitamin is vitamin B6, that is pyridoxine. So pyridoxal phosphate, okay, pyridoxal phosphate activates delta amino leolinic acid synthase enzyme, which catalyze the first reaction of heme synthesis, that is succinyl CoA plus glycine. Okay, first reaction, that is succinyl CoA with glycine, that is catalyzed by ALA synthase, delta amino leolinic acid synthase, and this ALA synthase require vitamin B6, that is pyridoxal phosphate. Okay, this is third one. Fourth one is vitamin C. Now it acts as a reducing agent. It reduces ferric ion to ferrous ion, and in the ferrous ion, iron is absorbed from the intestine. So ferric to ferrous, conversion of ferric form of iron to ferrous form of iron, it is done by vitamin C. It's required. Okay, so vitamin C, again you require acidic environment there also. So in addition to acidic environment, you require vitamin C, which reduces ferric ion to ferrous ion. So enhances the iron absorption. So these are enhances the iron absorption from the intestine. So these are the main four hematopoietic vitamins. Okay. Now I have enumerated of these four. In addition to this, there is another fifth one, that is pantothenic acid. How it is a hematopoietic vitamin? It is a constituent of coenzyme A. Okay. What is the role of coenzyme A? It binds with the succinate to form succinyl CoA, which then enters the heme synthesis pathway. Okay, so first step of heme synthesis you must be able to recall, that is succinyl CoA plus glycine. Okay, so formation of succinyl CoA require pantothenic acid because it is the constituent of coenzyme A, which binds with the succinate and forms succinyl CoA, which can get channeled for heme synthesis. Okay, so these are the five hematopoietic vitamin. Okay, so it is of 2 marks, so you can write any four. The first four are very important as your hematopoietic function is concerned. First four: folic acid, vitamin B12, vitamin B6, and vitamin C. Okay, so these are the hematopoietic vitamin. I am making a separate video on hematopoietic vitamin. Okay, so there the role of each vitamin is answered. So I will explain the role of each vitamin, each hematopoietic vitamin, what is the exact role, what is the exact reaction it catalyzes. Okay, so separate video will be posted on hematopoietic vitamins. Okay, so this is only of 2 marks, so I have just provided the ideal solution. Enumerate 4 vitamins and their function, how they are acting as hematopoietic vitamins. Okay, let's see the next question. 2G, that is draw electrophoretic pattern of any two disease. Okay, very easy question. My suggestion will be, before drawing electrophoretic pattern of diseases, you must draw the electrophoretic pattern in normal scenario. Normal electrophoretic pattern you must draw, because Based on that, the examiner will be able to identify the differences between normal and the abnormal. Okay, so first will be your first just draw this pattern, normal pattern. Okay, now, while drawing electrophoretic pattern, you must draw the point of application, anode, cathode, and different bands, which are albumin, alpha 1, alpha 2, beta, and gamma. Okay, this is the normal pattern. Now draw the abnormal pattern. Now, while selecting the diseases, I have selected the two which are easy to remember and easy to draw. First one is nephrotic syndrome, where albumin and most of the protein get lost through the urine. Okay, but of large proteins like macroglobulin, alpha 2 macroglobulin, they are not able to excrete through the glomerulus. So that concentration of alpha 2 band is increased, and as albumin get lost, the concentration is decreased. So band is less here as compared to normal. Okay, less band of albumin, density is also decrease, and alpha 2 band get widened. Okay, wide alpha 2 band, because alpha 2 band majorly is formed by alpha 2 microglobulin. So that is why alpha 2 band is wide here in nephrotic syndrome. Second example I have taken is a multiple myeloma, because all of you know there is a M band. But where is an M band? That is between beta and gamma region. Okay, this is called as M band. Okay, so these are the pattern in diseases. So these are easy to remember. Nephrotic syndrome, you know, protein get lost in the urine, so albumin band is decreased, and alpha 2 macroglobin is increased, so alpha 2 band is wider. Okay, in multiple myeloma, you know the M band. M band is there, but you should know the location. It is between beta and gamma region. Okay, let's see the next question. Question number 2H, write daily requirement of proteins with two examples of complete protein. Okay, daily requirement along with the example of complete protein. Daily requirement is recommended dietary allowance. What is RDA for protein is 0.75 gram per kg body weight per day. Okay, 0.75 gram per kg body weight per day. This unit is very important. Okay, per gram, 0.75 gram per kg body weight per day. And what are the examples of complete protein? Complete protein means a protein that contains all the 10 essential amino acids. So what are the two complete protein? First is casein of milk, and second is egg albumin. Egg albumin and casein of milk. These are the examples of complete protein. Okay, so very straightforward question. 1 mark for RDA, 1 mark for examples of complete protein. Let's move to the next question. Write composition of oral rehydration solution. Okay, so most of the book do not have this composition of ORS, but only most of the book mention that in the treatment of diarrhea, this ORS is used. What is the composition? First component is sodium chloride. So you have to write the name of the component and concentration in gram per liter. Again, in biochemistry, unit is very important. Whether you are doing your practicals or whether you are answering in the theory paper. So sodium chloride concentration is 2.6 gram per liter. Next is potassium chloride 1.5 gram per liter. Anhydrous glucose 13.5 gram per liter, and trisodium citrate dihydrate 2.9 gram per liter. Okay, so there are four components: sodium chloride, potassium chloride, anhydrous glucose, and trisodium citrate dihydrate. Okay, so you have to write component along with the concentration. Okay, so this is the composition of oral rehydration solution. Let's move the subsequent question. Write any two biochemical parameters derange in starvation. Very easy. Okay, write any two biochemical parameters which are derange in starvation. So just apply your common sense. In starvation, what will happen? Glucose will decrease, your insulin will decrease, glucagon will increase acetyl CoA, increase ketone body formation, and decrease glycogen. So I have provided 6 parameters. You can add your other parameters as well, but these are the important by and easy to remember, because in starvation obviously glucose will decrease. Okay, obviously your glucagon will increase and insulin will decrease. There will be the formation of ketone body. So first acetyl CoA increase, which leads to increase ketone body formation, and glycogen is getting broken down, so glycogen level will decrease. So these are the biochemical parameters which are deranged in starvation. Okay. Lastly, enumerate 4 causes of metabolic acidosis. Again, a straightforward question. Write 4 causes, get 2 marks. Okay, I have divided this causes of metabolic acidosis into two. That is normal anion gap metabolic acidosis and high anion gap metabolic acidosis. So high anion gap metabolic acidosis, which are the causes? Lactic acidosis, okay, diabetic ketoacidosis, prolonged starvation, renal failure, and methanol poisoning. Okay, so these are the causes of high anion gap metabolic acidosis. Now, what are the causes of normal anion gap metabolic acidosis? Severe diarrhea and renal tubular acidosis. Severe diarrhea and renal tubular acidosis. So, lactic acidosis can be due to various causes like in cancer, emphysema, cyanide poisoning, carbon monoxide poisoning. Okay, then when in diabetic ketoacidosis and prolonged starvation also there will be the metabolic acidosis, renal failure, and methanol poisoning. Renal failure and methanol poisoning. So lactic acidosis, diabetic ketoacidosis, prolonged starvation, renal failure, methanol poisoning. High anion gap metabolic acidosis normally, and for normal anion gap metabolic acidosis, severe diarrhea and renal tubular acidosis. Again, these are asked in the MCQ as well. Okay, so this was the last question. Thank you.